Okay, so let me see. So John and Sankar spoke about the, well, let me share my screen. Where's Zoom gone to? Oh, come on. Okay, can you see my screen? Yes.
Yes, we can.
Fine.
Okay. So John Shankar spoke about the Metabolomics Omic browser yesterday, and Shankar mentioned it very kindly this morning. So in this lightning talk, I'm going to describe in more detail some of the interactive features of the Metabolomics Omic browser, specifically NGCHMs. We believe the interactive features of Metabolomics Omic browser are important for the detective work behind identifying batch effects and other interesting features in the data. We pull data regularly from the Metabolomics workbench via an API, kindly provided by Shankar and Owen. Then after we've completed our analysis, MWB then incorporates pointers to our analysis for the analyzed studies. If you want to find Metabolomics Omic browser, one way to do that is to go to the Metabolomics workbench study page and click on statistical analyses. Then you can scroll down the page until you come to the sections on the Metabolomics Omic browser and click on the link to a particular analysis, and then it will take you to the MOB browser in a view like this. So now I'm going to switch over to the interactive view of the system. And since I was playing around with it, it wasn't where I wanted it to be. But the default visualization is a PCA plus plot. And you can see that you can highlight points and interact with it if you want. If you click on the algorithm page, then you can go to a variety of other visualizations. Just to note, one of those is UMAP, which provides an alternative low-dimensional visualization of the data. And another one is next generation clustered heat maps, which I'm going to spend most of the time on. First of all, I'm going to sort of increase the amount of space that's available for looking at the NGCHM by hiding that panel and minimizing that panel. And I'm going to sort of move this over a little bit because you don't need quite as much room for that. Don't move it too fast. And I'm going to make room for other stuff by shrinking the summary view down a little bit. So one of the new features that we've added is the ability to add additional panels into the view for displaying other heat maps or other visualizations. One use for it is to display another detailed heat map. So now we have two detailed heat maps, and we can use that to sort of move the second one around and come to a different view. So one use of that is the ability to look at two different parts, two widely separated different parts of the heat map, and look at them simultaneously. Another thing we can use is to display other sorts of interactive graphs. And one of these is the scatter plot, which is actually provided by a completely separate tool that communicates via an API with the NGCHM. I'm going to select some coordinates to display. I'm going to base these on data from the heat map. I'm going to select that bunch of genes, and in the middle of that is oleic acid. So I'm going to call them all by that because I'm not a biologist and that's just what I know. And then I'm going to select the set of genes below that and grab those as well as the second axis. And they contain glutamate. So I'm going to make those glutamate for want of a better name. And then I'm going to sort of generate the plot. Once it's made, you can sort of increase the size of the points so that they're visible and that you can work with them. And one of the things you can do with these now that you've generated is sort of zoom and pan it just like you would zooming and panning within the NGCHM. But perhaps a more interesting thing is you can change into lasso mode, and that allows you to interactively select points within the scatter plot. And you'll notice that when I did that, not only were the points in the scatter plot selected, but also the same points were selected in the NGCHM, both in the summary view and in the detail view. And the browser works to maintain a single user visible view of what is the current selection across the NGCHM and all the plugins that are selected in it. We can see this also by going back to the other direction, and we can, for example, go and select all the points whose diagnosis, this is how I list it, is control, and selecting those, and we select those points in the NGCHM, and they're also selected in the scatter plot via the highlights around the individual data points. Another tool that we have integrated at the moment is our Pathway Mapper tool, and that lets us display a network diagram or pathway diagram. And it should be able to display data inside these pathway diagrams taken from the NGCHM, but that's not going to work because this is a gene network and this is a metabolomics metabolite heat map. So I'll switch back to the PowerPoint, and this is an example of what it might look like if we're taking data directly from a heat map into the pathway mapper. We're in the process of developing detailed new pathway diagrams. This is an example of one of the diagrams that we're developing. It contains both metabolites and proteins. And we're also in the process of developing a new pathway visualization tool that's based on the Newt pathway editor, viewer, and editor. And it's going to use a standard pathway format called systems biological graphical notation. We'll be able to import pathways either from our own database of built-in or pathway server database, as well as other pathway network tools and databases, as long as they provide the data in that format. Going forward, we expect to maintain integration with the Metabolomics Workbench toolset and potentially other metabolomics tools. And please feel free to contact us to discuss integration or interoperability ideas. Thank you very much. Happy to take questions in the chat.
Great. Okay, so we can